Good morning, good afternoon, and good evening from wherever you're joining us today. My name is Wasundara Fernando, representing the CUFSAA, and I'll be the moderator for today's talk. First of all, on behalf of CUFSAA, Distinguished Speaker Series Subcommittee, I warmly welcome you all for today's talk. I'd like to remind you that this talk will be broadcasted on our Facebook page and YouTube channel simultaneously. There will be a Q&A session, and I invite you to type your questions in the Zoom chat or on our Facebook comment section at any time, and we will direct the questions to our speaker at the end of the talk. Today, we have Professor David Hoskin, and he's going to tell us about his research on iron withdrawal by selective chelators for cancer treatment. Dr. David Hoskin is Professor Emeritus in the Department of Pathology at Dalhousie University. He is also a lifetime honorary member and senior scientist of the Beatrice Cancer Research Institute, as well as the inaugural holder of the Canadian Cancer Society, Canadian Breast Cancer Foundation, Atlantic Region Endowed Chair in Breast Cancer Research. Professor David Hoskin graduated with his B.Sc and Ph.D. degrees from McGill University and completed his postdoctoral training at the Samuel Lunenfeld Research Institute at the University of Toronto prior to accepting an academic position at Dalhousie University. Professor Hoskin's publications include over 200 research and review articles detailing his studies on the anti-cancer and anti-inflammatory potential of natural source compounds, including antimicrobial peptides and phytochemicals found in common spices, fruits, and vegetables, and the manipulation of T lymphocyte signal transduction using natural source compounds and other novel agents for cancer control. The focus of Dr. Hoskin's research career was the development of new treatments for breast cancer and other common cancers. Although retired, Professor Hoskin remains involved in teaching and cancer research at Dalhousie University. He is also an active member of the Royal Astronomical Society of Canada and serves on the board of directors for the Halifax Centre. On a personal note, I'm honored to have had the opportunity to complete my Ph.D. under Professor Hoskin's supervision, and today to introduce Professor Hoskin to deliver a talk at our distinguished speaker series. Without further ado, Professor Hoskin, with great pleasure, I invite you to tell us about iron withdrawal by selective chelators for cancer treatment.
Okay. Can everyone see that?
Yeah, thanks.
Yeah, thanks, yes.
Okay, perfect. So I'll begin by thanking the organizers, and particularly Dr. Fernando, for the invitation to speak at this seminar series. As Dr. Fernando mentioned, I'm going to detail some work that was done in my lab looking at the potential of using iron chelators as a cancer treatment, either alone or in combination with chemotherapy and radiation therapy. So iron is a key cofactor in a number of physiologic processes. It's essential for respiration, for oxygen transport, cell proliferation, metabolism, detoxification, but it can also be potentially toxic. So if there's not enough iron that available for cells, that impairs the activity of a wide range of iron-dependent proteins, and that leads to cellular dysfunction and disease, which is treated by iron supplementation. On the other hand, excess iron can also be dangerous because at high levels, iron promotes the generation of free radicals and other reactive intermediates that cause oxidative stress and damage cells and more importantly, damage DNA, leading to mutations and different diseases such as cancer or arteriosclerosis. So ideally, one wants a balance to be in place in terms of how much iron is in a cell. One of the most, I guess, dangerous aspects of iron overload is that it can lead to the production of these reactive oxygen species through Fenton chemistry. And as I said, that causes damage to different cellular components. Because of this need to maintain an appropriate level of iron, the intracellular levels of iron are regulated by various uptake and storage mechanisms. In cell growth, iron is a key component. It's essential for the functioning of many enzymes, including ribonucleotide reductase that's involved in DNA synthesis and repair, as well as a number of other DNA repair enzymes. Iron also affects gene expression by regulating transcription factors like NF-kappa B and HIF-1 alpha that in turn induce the expression of genes that promote cell survival. And as I said, it catalyzes the production of reactive oxygen species. Now, at low levels, reactive oxygen species are important signaling molecules because they induce the generation of extracellular regulated kinase 1 and 2 signaling, which is a survival promoting mechanism for cells. So it's only at high levels, that very high levels, that one sees the damage to cellular components by reactive oxygen species. This slide shows the mechanisms by which iron is regulated, its uptake and its excretion are regulated in cells. The primary mechanism for iron uptake by a cell is via transferrin receptor type 1, which interacts with transferrin that has bound ferric iron ions, creating diferric transferrin. When that diferric transferrin interacts with the transferrin receptor 1 molecule, the complex is internalized by endocytosis. And then within the endosome, the ferric iron ions, the trivalent ions, are released and converted to ferrous iron, which is then transported via a molecule called DMT1 into the labile iron pool of the cell. Now, some of that iron is then stored in ferritin, whereas other iron molecules within the pool are utilized in mitochondria for energy generation and for activities such as DNA synthesis and repair, and the effects of necessary actions of other cellular systems. Excess iron is exported from cells by a membrane-associated molecule called ferroportin, and the synthesis of ferroportin is in turn regulated by hepcidin. So if there is reduced availability of iron, the cell will want to downregulate ferroportin to maintain its labile iron pools, and that process is mediated by the expression of the hepcidin molecule. Now, iron has an important role in cancer because the rapid proliferation of cancer cells means that they have an increased need for iron for these DNA replication and repair molecules compared to normal cells. In addition, many cancer cells alter their iron import machinery so that they can take up more iron. For example, cancer cells typically show increased expressions of transferrin receptor 1, and at the same time, a decreased expression of ferroportin. So not only do the cancer cells take in more iron, but they also restrict its export from the cell. And as I mentioned, iron through the Fenton reaction can cause different levels of oxidative stress depending on the abundance of iron. At low levels of oxidative stress, you get these induction of signaling pathways involved in cell proliferation, which promote growth of the tumor and also progression because of the mutagenic effects of intermediate levels of reactive oxygen species on DNA. And some of those mutations result in the cancer cells becoming more aggressive. Now, at very high levels, oxidative stress is cytotoxic even to cancer cells and can result in apoptosis or necrosis of the cells. So this is a slide which details some of the differences between normal epithelial cells, for example, and a transformed or malignant epithelial cell. You can see here the transferrin molecule, transferrin receptor molecule interacting with transferrin that's been loaded with ferric iron and expression of the iron exporting ferroportin, and there's little or no hepcidin produced under these conditions. In contrast, in a malignant cell, expression of the transferrin receptor molecules is increased so that the cell can take in more iron, increasing the size of its labile iron pool. And at the same time, hepcidin is generated to downregulate the expression of ferroportin so that the cancer cell has access to the maximum amount of iron within the labile iron pool. In addition, in breast cancer in particular, iron can play other roles. For example, during redox cycling of estrogen, which is an important molecule in supporting the growth of certain breast cancers, when redox cycling of estrogen to semiquinones and quinones takes place, superoxide ions are produced, and ferrous iron can then interact with hydrogen peroxide to form additional hydroxyl radicals, which in turn causes oxidative stress, DNA damage, and mutation, and promoting the development and progression of breast cancer. So this dependence of cancer cells on the availability of iron for their growth and the mutation-induced progression suggests that removal of iron using different iron chelators might be a viable strategy to suppress or prevent tumor growth or tumor progression. There are several iron chelators that are currently in clinical use to treat iron overload in patients, which, as I said, is toxic. These iron chelators that are in clinical use are deferoxamine, deferiprone, and deferox. They have different structures. Deferoxamine is derived from a modification of a bacterial sideramine, Diferiprone is a synthetic hydroxypyridone class chelator that is a fairly small molecule, so it can enter cells, as is deferasirox, another small lipophilic molecule that can enter cells. In contrast, deferoxamine is larger and doesn't enter the cell, so it remains extracellular. The iron chelator that my lab use is a molecule called DIBI. This is a novel, highly selective iron chelator that was developed by a biotechnology company called the Chelation Partners Incorporated. And they developed this molecule as a antimicrobial agent, but upon further investigation, it became apparent that DIBI was also potentially useful as an anti-cancer agent. It's an interesting molecule. It's rather large. It consists of a number of hydroxypyridinone chelators attached to a polyvinyl pyrrolidone copolymer backbone. And the molecules that actually interact with ferric iron are MAHMP molecules. And you can see that there are a number of these MAHMP molecules distributed along the copolymer backbone. And that allows for a very strong avidity for individual trivalent ferric iron ions and increased ability over some of these other chelators to actually chelate extracellular iron. In addition, different toxicity profiling studies demonstrated that DIBI is completely non-toxic, whereas these three iron chelators that are in current clinical use have different toxicities that sometimes restrict their use. So the first question to ask is, well, how good is DIBI on a molecule per molecule basis compared to some of these other iron chelators? And in this assay, we looked at the ability of DIBI and other iron chelators to restrict the growth of different cancer cell lines. And it's apparent here that the EC50 of DIBI for 50% inhibition of the growth of a breast cancer cell line is much lower than that of diferiprone and roughly equivalent to that of deferoxamine. And of course, as expected, is more effective than MAHMP alone because of the multiple MAHMP residues on the copolymer backbone of DIBI. In certain cancer cells, such as this murine fibrosarcoma line, DIBI was more effective than any of the other chelating compounds. So, DIBI was certainly showed a lot of promise as a viable alternative to some of the clinically available iron chelators for the treatment of cancer. One of the first things that we looked at was comparing the effect of DIBI versus MAHMP alone on cancer cell growth. And we're looking at a breast cancer cell line over a period of 72 hours after the addition of DIBI or MAHMP to chelate extracellular iron. And as we would expect, DIBI is much more effective, virtually stopping all metabolic activity in the cancer cell culture at 50 micromolar concentration, whereas the same concentration of MHMP alone was having little effect. DIBI is also very selective in its effects on breast cancer cell growth. And this slide details a few experiments that demonstrate that selectivity, the fact that the reason that DIBI inhibits cancer cell growth is because of its ability to restrict iron availability to the growing cancer cells. On the left, you see data from an experiment where cells were loaded with calcium AM, and this molecule within cells becomes fluorescent, and that fluorescence can be quenched by intracellular labile iron. That means then that if iron levels are diminishing within the cell, the fluorescence of the calcium will be increased. And indeed, that's what's shown here. Unstained cells, so cells that haven't been treated with the calcium AM, in the presence of DIBI alone have no fluorescence. When you stain those cells and increase the concentration of DIBI and thereby restrict the availability of iron and leading to decreased intracellular labile iron, there's a dramatic and significant increase in the fluorescence of those cells. So that's consistent with a reduction in the labile iron pool by DIBI-treated cancer cells. In addition, if one adds iron back into a DIBI-treated cancer cell culture such that there's now excess iron available, so you've overwhelmed the ability of DIBI to chelate the available iron, that mitigates the inhibitory effect of DIBI on cancer cell growth. The inhibition of breast cancer cell growth by DIBI-mediated iron withdrawal is associated with the accumulation of the cancer cells in the S phase of the cell cycle, as shown here. So there's a significant increase in the percentage of cells in S phase of the cycle in both MDA-MB 468 human breast cancer cells and 4T1 mouse mammary carcinoma cells. That increase in the number of cells, the percentage of cells in the S phase, is consistent with a decrease in the percentage of cells in the G1 phase of the cell cycle. Cells that become arrested in the S phase of the cell cycle exhibit extensive DNA damage. And that's shown here. We're measuring the fluorescence of breast cancer cells stained with a DNA damage marker, gamma H2AX, which is a phosphoprotein required to recruit DNA repair proteins to the nucleus where they repair the damaged DNA. So increased binding of antibodies to gamma H2AX, these are fluorescent antibodies, is a well-established marker for DNA damage in the form of double-stranded DNA breaks. Here we're looking at control breast cancer cells and gating on the part of the cells in the S phase of the cell cycle. And when you can see that when DIBI is introduced, there's an increase in the expression of this DNA damage marker within cells in the S phase of the cycle. This could also be seen in these fluorescence microgram photographs showing that we see no gamma H2AX expression in the control cells, whereas following DIBI treatment, there's a lot of gamma H2AX being expressed. And in addition, we also see this molecule phospho-CHEK2, which is another marker of DNA damage being upregulated in DIBI-treated cells compared to the untreated control cells. One would expect then that if iron withdrawal by DIBI is promoting DNA damage and the accumulation of damaged cells in the S phase of the cell cycle, that chemotherapeutic agents that also function by causing DNA damage might in fact show increased activity when cells are treated with DIBI. And indeed, that's what we found. Compared to treatment with cisplatin or doxorubicin alone, the EC50 shown here for that drug is decreased quite dramatically when those cells are first pre-treated with DIBI to limit the availability of iron to those breast cancer cells. So in the case of a fairly small amount of DIBI, 0.2 milligrams per mil, we've got a tenfold decrease in the EC50 of cisplatin required to have a cytotoxic effect. This means that the combination of an iron chelator such as DIBI with chemotherapeutic drug might allow that lower concentrations of that drug to be used in cancer treatment, and therefore limit the toxicities that are associated with chemotherapy in cancer patients. So we thought this is quite an exciting finding. It follows also that since ionizing radiation acts by damaging DNA, then in a similar fashion, if you restrict the availability of iron to a cancer cell subjected to ionizing radiation, that cell, because of the need for iron in DNA repair, that cell would not be able to repair its damaged DNA. And in fact, that's the case. So when we subjected breast cancer cells to 6 gray of ionizing gamma radiation, shown by the plus signs, that effect was increased in the presence of DIBI. So there's a significant increase in the percentage of dead cells when treated with DIBI plus ionizing radiation compared to ionizing radiation alone. Another aspect, so far I've been talking about iron chelation as a potential mechanism to treat established tumors, but cancer development might also be a suitable target for iron chelators. because it's well established now that chronic inflammation is one of the enabling factors for tumorigenesis. So inflammation promotes tumor development. In large part, this is because of the action of professional phagocytes like macrophages that are stimulated during chronic inflammation, and those macrophages then secrete different inflammation promoting cytokines as well as reactive oxygen and nitrogen intermediates. So we therefore asked whether limiting the availability of iron might impact inflammatory responses. This is a schematic which illustrates the interaction between iron and redox-sensitive transcription factors, pro-inflammatory cytokines, and reactive intermediates. So as there's an increase in the availability of iron within a cell, that causes increased activation of transcription factors like NF-kappa B, leading to the synthesis of pro-inflammatory cytokines such as interleukin-6, that in turn lead to the sustained activation of immune cells, including macrophages, leading to the expression of reactive intermediates, which then further induce the transcription factor activation. So it becomes an amplifying cycle. And of course, once these reactive intermediates reach a certain level, one sees damage to DNA, protein, and to lipids. So that's all associated with inflammation. It turns out that, as we would predict, the withdrawal of iron from cultures of macrophages that have been stimulated with lipopolysaccharide as an inflammation inducing stimulus, those macrophages produce less interleukin-6 and less interleukin-1 beta than control cells. And we've also here looked at the effect of the copolymer backbone alone on the action of these macrophages in response to lipopolysaccharide, and there is no effect. Again, in this case, the suppression of interleukin-6 and interleukin-1 beta by iron withdrawal is unique to the intact DIBI molecule. Now, very curious here, we were unable to show any impact of iron withdrawal at these levels on the expression of TNF-alpha, tumor necrosis factor alpha, which is another key pro-inflammatory cytokine. So the effect of iron withdrawal with DIBI isn't global in terms of macrophage function. And that also, this lack of effect on TNF-alpha expression suggests that DIBI is not affecting the activation of NF-kappa B transcription factor. And in fact, when we looked at that, we saw no evidence of iron withdrawal by DIBI suppressing NF-kappa B activity. What we did find was a decrease in the ability of certain STAT molecules to signal. And these STAT molecules, STAT1 and STAT3, are involved in amplifying the expression of pro-inflammatory cytokines such as interleukin-6. Iron withdrawal by Deepthi also suppresses the ability of LPS stimulated macrophages to produce reactive intermediates. So the accumulation of reactive oxygen species in response to LPS, shown here, is suppressed in the presence of Deepthi, as is the expression of nitric oxide, which is a precursor for the development of different reactive nitrogen species. So this is all consistent with a anti-inflammatory response by iron chelation on macrophages. So this is the schematic then summarizing the anti-inflammatory action of iron withdrawal by iron chelators such as Deepthi, macrophages that are important in inflammatory responses, and also are a poor prognostic factor in different cancers. So if there are a lot of macrophages infiltrating a solid tumor, that tumor then has a poor prognosis, most likely because of the mutagenic and cancer progression promoting activities of reactive oxygen and nitrogen intermediates. The impact of iron chelation on anti-cancer activity of different compounds extends beyond conventional pharmaceutical agents that chelate iron, and includes, in fact, a number of phytochemicals. So curcumin is a principal curcuminoid of the spice turmeric. It has a long history of use in traditional Asian medicine to treat a range of different illnesses. And more recently, preclinical studies have shown that curcumin can inhibit multiple pro-inflammatory signaling pathways, so it has an anti-inflammatory activity. Therefore, perhaps able to prevent carcinogenesis. And a number of laboratory and animal research studies show that curcumin also has potent anti-cancer activities. Unfortunately, its use clinically has not been very successful due to its inherent chemical instability and poor bioavailability. But nevertheless, in laboratory studies, curcumin does have potent anti-cancer activity. My lab studied curcumin a number of years ago, and here we showed that, and this was looking at a human colon cancer cell line, absorbance indicating cell viability. So high absorbance in an MTT assay indicates lots of cells. A reduction in absorbance indicates that cell death, so fewer cells in culture. And one sees a nice dose-dependent inhibitory effect on cell viability, regardless of whether these cells express p53 or not. Now, how does this come back to iron chelation? Well, turns out that curcumin is a rather good chelator of iron. So at least some of the anti-cancer activities of curcumin and other iron chelating phytochemicals are likely due to its ability to remove available iron and prevent it from being utilized by cancer cells. So just to summarize this part of the seminar, cancer cells, due to their rapid cell growth, have an increased need for iron. As a result, if iron is withdrawn, cancer cell growth is reduced due to the induction of S-phase cell cycle arrest and DNA damage that's consistent with apoptosis. Iron withdrawal also is able to sensitize cancer cells to different chemotherapeutic drugs and ionizing radiation, which means that lower doses of these drugs and ionizing radiation may be able to be used in combination with an iron chelating agent to get the same effect as increased doses of these drugs and radiation, which can also have adverse side effects. When iron is limited, inflammatory responses are suppressed, and that means that both cancer development and the progression of tumors would be inhibited by the presence of an iron chelator. And finally, iron chelation may contribute to the anti-cancer activity of certain phytochemicals. Now, the organizers asked me to talk a little bit about my hobby, which is amateur astronomy and astrophotography. And it's actually a little bit linked to my research on iron and its role in cancer cell development and progression. Iron, and indeed all elements, are made within stars. As stars age, fusion reactions within the star produce different elements, and one of which is iron. That star then, when it dies, either exploding in a supernova or releasing most of its mass as a planetary nebula, those elements are released into space. So all of us are made of elements that were originally produced in a star. So this is an image of the Andromeda galaxy containing many stars, a number of which are supergiants nearing the end of their lifespan, and therefore a potential source of elements such as iron. This is one of the telescope rigs that I use to take pictures, such as this. Stars are born within clouds of hydrogen gas and dust. These are called emission nebula. This is a nebula called the Lagoon Nebula, located in the constellation Sagittarius. And you can see here a cluster of newly formed stars, that some of which will become supergiant stars and result in the production of all of the elements in the periodic table, and eventually being used to create planets and ultimately one would expect life forms. This is another emission nebula called the Trifid Nebula. When stars go supernova and release those elements that have been created within them, they leave a supernova remnant. This is the Crab Nebula, located in the constellation Taurus, which is a supernova, which is the remnant of a supernova that was seen by Chinese astronomers over 1,000 years ago. Now, not all stars explode in a supernova. Some release most of their mass as a cloud of gas, and these are stars that form planetary nebulas. This is an image of the Dumbbell Nebula. So you can see here a very faint star at the center. This is the white dwarf, which was the source of all these gases that are forming this very nice looking, very beautiful nebula. We find iron distributed throughout the solar system. In this picture of the colorized picture of the moon actually has colors. And the reddish hues are the result of iron-bearing minerals on the surface of the moon. The blue hue is the result of titanium-bearing minerals. Now, we don't have to worry about our own sun going supernova. It's not large enough, but it is very active. This is a hydrogen alpha image of our nearest star that I took a couple of months ago, showing the solar prominence, so gaseous plasma being ejected into space from the surface of the sun. And here is a sunspot complex about to rotate out of view. So with that, I'll finish and acknowledge the hard work of a number of individuals in my lab involved in this research: Dan Arsenault, Melanie Coombs, Wasundara Fernando, Javed Gassemi-Rad, Taryn Grant, Anna Greenshields, and Alicia Malone. Chelation Partners provided us with the iron chelating compound Bibby, and additional funding for the experiments came from MITAX, the Faculty of Medicine, NSERC, and the Canadian Breast Cancer Foundation. So I'll end, and I'm happy to answer any questions. Thank you.
Thank you, Professor Hoskin, for the wonderful talk. It is open for questions now.
Thank you, Professor Hoskin, for that brilliant talk. I am very interested in what you said, because they're really fascinating for me, because in our lab, we also work on anti-cancer properties, but of natural products, especially from Sri Lankan traditional medicine, taking cues from Sri Lankan traditional medicine. My question, I have a few questions, but the first one is, so is this a global phenomenon that is, applicable to all carcinoma cells, Prof? Because you said sarcoma, it acts very well on sarcomas.
Yeah.
And you did mention all kinds of different types of cancer cells, breast cancer, and so on.
Yeah,
so
it's generally a global effect. So all malignant cells require more iron than their healthy counterparts.
So this is eye-opener for us too. I mean, we do work on different aspects, but these two, and I was really surprised to see curcumin that we all know about, and that maybe it is established now. Is that it, sir? That it is an iron chelator. Is that?
Yeah, it's pretty well established, and there are other natural products, phytochemicals, that based on their structure are likely to be able to chelate iron. Piperin is another molecule from black pepper that my lab studied, and I showed the structure of piperin to my collaborator at Chelation Partners, and he felt that it would likely also chelate iron. So I think it's worth showing the structure of the compounds that you're working with to a medicinal chemist, and see whether there might be an aspect of iron chelation involved in their activity. An easy way to test that would be simply to add extra iron back into the culture and see if you can decrease the anti-cancer activity of the compound that you're studying.
Thank you so much. Thank you. I really will pick your brains on that. And so what next, Prof? So this beautiful work now, what are you going to do next? What can be? I mean, brilliant, and you know, I'm just curious.
Yeah, well, my understanding is that Chelation Partners is now pursuing preclinical or clinical trials on DIBI, not for cancer anti-cancer activities, but for its antimicrobial activities, which was what they originally developed it for. And I suspect that if they're able to get it, the compound into clinical trials for drug-resistant bacterial infections, and it's shown to be safe, then they will look into to pursuing its potential use in cancer treatment.
Thank you, Prof. All the best. That was lovely.
Dr. Hoskin, we have a few other questions as well.
Sure.
Is there a possibility of removing other important biological metals by DIBI? One of the questions.
No, DIBI is very selective for iron. Unlike some of the other iron chelators that also chelate copper and aluminum, for example. So DIBI would only be used in situations where iron is important, as for the growth of prokaryotic cells like bacteria and eukaryotic cells, in particular transformed cells. You're frozen, Wasu.
Frozen.
Yes, I think Wasundara got disconnected. Let's give a minute whether she will join again. Yes, so there is another question. So can AI be used to accelerate this line of research? Is it already being used?
I'm not sure what you mean by AI.
I think the artificial intelligence, or like computing neural net. I think, yeah, probably.
Yeah, so, sort of in silico experiments. I imagine that one could use that to discover other potential iron chelating compounds, but I don't know if that's whether that's actually being used currently.
Okay.
I know it's used in drug discovery quite extensively now.
Right. Okay. Let's see. I don't know whether Professor Udagama already asked this question, but does DIBI cause any liver or kidney toxicity?
Not that the studies Chelation Partners had performed detected. So it seemed to be remarkably non-toxic, either given orally or intravenously.
Okay.
And now, of course, this was in a rat study, right? So it's always conceivable that with human beings, you might have a toxic effect that you don't see in rats, but certainly rats tolerated very high doses of DIBI.
Okay. Wasundara, you are back. So I asked few questions.
Sorry, there was a technical difficulties. Did you ask all the questions?
Well, I asked number 3 from our talk and also what Niranjak asked on the chat.
Okay, good. I think that's all the questions we have.
Great.
Yeah.
Thank you, Dr. Hoskin, again for the talk.
No, thank you. Thank you for the invitation, Dr. Fernando. And I look forward to seeing you back in Canada at some point.
Yes, soon, hopefully. Okay, so we would like to hear from you. Please let us know your suggestions and comments by contacting the CUFSAA Speaker Series Subcommittee using the email on the slide. Also, let us know if you have any specific suggestions for topics and speakers. Here is a quick peek into the next 2 months. Dr. Vinod De Silva, the Vice President, the lead discovery and optimization, small molecule drug discovery in Bristol Myers Squibb, will be with us next month giving a talk, and join us to listen to Professor Rangadi Dias, Assistant Professor from University of Rochester in November. Now I would like to invite Professor Udagama, Head of the Department of Zoology and Environmental Sciences, University of Colombo, to deliver a vote of thanks for today.
Right, so hello everybody, and greetings from the Faculty of Science, University of Colombo, specifically from the Department of Zoology and Environmental Sciences, because I think we are the closest to the departments touching on this subject. So for me personally, it was a fascinating talk. Thank you so much, Professor Hoskin. It was actually a privilege to listen to you and to see all these fantastic data that you have generated. And congratulations, Wasundara, as you have been part of this. So I too, you are from Dalhousie. I spent 4.5 years at UNB, University of New Brunswick, on an insert fellowship, my postdoc. So I know, Professor Hoskin, how lovely Canadians are, and you know, I have really nice memories of Canada. And so this is to profusely thank you for taking, you know, off your very busy schedule. You must be extremely busy doing all this fantastic work, and to be with us this morning. For us, it's night, and I'm in Sari, in case others are wondering. I just got back from work, 5 minutes before the talk started. So I'm sorry that the students are not there in numbers, because this is a really bad week. This is the last week of our semester, and they have assignments and assessments and tutorials, and everybody's rushing to complete their lectures and so on. But that's not an excuse. So we were the losers, you know, not being there in numbers. So I wish you all the best, and to carry on with this fantastic work, and I hope your molecule will, eventually be extremely useful to humankind. And I would also like to take this opportunity to profusely thank CUFSAA, because CUFSAA is like Santa Claus to us at the Faculty of Science, University of Colombo. We have been very fortunate, especially during the last couple of difficult years, and more so now, because they have been supporting us through various measures, giving us instruments, computers for the students, and a lot of other things. And I wish you courage and strength to go on doing this good work for us, because right now, never than before, we really need outside support. And so I also would like to thank the participants who are here, and of course, CUFSAA, the organizers, And I definitely do look forward to the next talk because that is something also in my area of expertise. And also, Professor Hoskin, I'm a basic immunologist. So, but you talked about, all these cells and cytokines and all those things are very clear to me and it was very fascinating. So thank you once again and hope everybody would have a good day there. And for us, we are at the last end of the day. So good night, everybody, for locals. The others, do please have a wonderful day. Thank you.
Okay, thank you for the invitation. Have a good day.
Thank you, Professor Udagama, for the lovely talk. And I just would like to remind that the talk is also available online on our Facebook page and YouTube channel for anyone who missed the talk today. Okay, so we have come to the end of today's seminar. Our next distinguished speaker is Dr. Bino De Silva, and we will announce the exact date and time of the talk in near future, so stay tuned. Thank you everyone for joining us today, and see you next month with another interesting seminar. Thank you.